So Clare Appavoo is the Executive Director of the Canadian Research Knowledge Network, which advances interconnected, sustainable access to the world's research and to Canada's documentary heritage content. CRKN is the administrative lead for the ORCID Canada and DataCite Canada consortia. Clare has recently been nominated to the board of ORCID. Clare, you can take it away.
Thank you very much, Christina, and I'm delighted to be here. I'm very pleased to introduce to you Peter Goodhand, who is the CEO of the Global Alliance for Genomics and Health, GA4GH, based at the Ontario Institute for Cancer Research in Toronto. He is also co-chair of the International 100K Cohort Consortium, IHCC, and a board member of RareX US, as well as being a board member on several Canadian and international advisory boards. I'm also pleased to introduce Dr. Guillaume Bourque, who is a professor in the Department of Human Genetics at McGill University. He is the Director of Bioinformatics at the McGill Genome Center and leads the Canadian Centre for Computational Genomics, C3G. He is also the chair of the Researcher Council of the New Digital Research Infrastructure Organization, fondly known as NDRIO. I'm very pleased to pass the mic over to you, Peter.
Thank you. Can I have my first slide, Christina? So, as you can see from my background in that slide, I am here speaking on behalf of the Global Alliance for Genomics and Health. We've been around. I've been working on it for 8 years. It's been up and running for about 7. But from the beginning, it was never just about genomic research. It was always about bringing genomics closer to healthcare and accelerating the science in a way that would really improve human healthcare. And we went for a global scale. So we went big from the beginning, and sharing data in Canada is difficult. Sharing data internationally can be really challenging. Next slide. From the beginning, led by Bartha Knoppers and her work out of McGill and CGP, we had a human rights foundation to what we were doing. And on these 2 fundamental human rights, the right to science and the right to recognition of work, particularly for scientists and others. Next slide. And that really was the underlying principles that went into our framework for the responsible sharing. This was our first big output. It's been modified, it's been updated, and it's been interpreted in different ways, both literally interpreted or translated into about 15 different languages, but also many of the components built out into more useful granular products and policies. Next slide. We've come from a world in which there were, we don't have to go back 2 decades to a world when there was only 1 human genome. It's a very recent mapping of the human genome that at a cost that was massive by any scale, $3 billion for the public project. Now we're less than $1,000, and it's moving out of the domain of a few elite centers into many, many universities, many healthcare settings. And we're talking about hundreds of individuals who will be sequenced at some level or another. Not everybody's going to get a whole genome. Not everybody needs a whole genome, but we are going to have hundreds of millions of genomic sequences in the not too distant future. Next slide. We're tracking at the moment over 200 genomic data initiatives. They could be generating genomic data, putting it in a repository, or they could be initiatives to take existing data and share it across many environments. Some of these are double counted, they'd be in 2 categories, but over 200 distinct genomic data initiatives globally. Next slide. Opportunity from a research perspective, and I think this has been, we're now starting to change our language a little bit because we're recognizing we need to address both the benefits to researchers of having secondary use of healthcare data, but we also need to address the benefits to clinicians and healthcare of having rapid access to genomic knowledge. So much more of a virtual.
Okay, I think we just lost Peter's feed for a second there. We'll just give it a moment and see if it comes back.
Me that's frozen?
Yeah, it's you that's frozen. I think
Okay.
you're back now. Thanks,
Okay.
Peter.
Okay. Next slide, please. The benefits of genomic data sharing, I'm not going to go through all. We'll make this slide deck available post the meeting. But in essence, depending where you're coming from in the research domain or the clinical domain or the policymaker domain, there are many benefits to the sharing of genomic data, particularly when you think about the diversity of human genomics. Next slide. But change in where the data has been generated and how it's been consumed means different models for it. So if the old model was somebody in an elite academic institute built up 500 genomes, and then other people downloaded it if it was publicly available, or they applied for permission to download the data, but essentially it was a repository, a few repositories, and people downloaded the data. Increasingly, we're seeing it's no longer viable, either from a scale of data or an ownership, a sovereignty of data, a privacy concern. So increasingly, we're changing the way we share data in a way that actually enables it to be more useful and to be better audited. But it's a very different approach than I built a big database and I'm going to decide who I let take my data. So I'll come next slide. So there's a paradigm shift from that data sharing in genomics no longer means data copying. It increasingly means data visiting. We leave the data in trusted repositories, safe havens, or where it was generated, and researchers go and visit that data, and ideally would do their compute, bring their algorithms to the data, and do it in a secure environment. And that is a great equalizer of access to data. However, it brings with it all kinds of other challenges. Next slide. And what we don't want to have to do is visit every one of those reference libraries one by one, learn different rules and different access. So it's really important, if we're going to move into a world where data visiting is the norm, that we can think about how best to federate search for the data, federated access to the data, federated analysis of the data. And so these two things tend to go hand in hand. Leave the data where it is, visit the data, but do it create as many approaches to interoperability and federation. And you can see how this influences the concept of ownership. And probably ownership of data in this context is less important than stewardship of that data. And there will be, whether the individual owns their data or the person who generated owns the data. The important thing is that we steward it and make it available. Next slide. And more than anything else, it's this shift of our need to link increasingly research data and health data. Health data has got none of the traditions that we've had in genomics of pulling of massive global collaboration. Healthcare data has always been siloed, always been local. In Canada, it's not even at a provincial level, it's a sub-provincial level. Just getting it to share from one side of University Avenue to the other is not easy, let alone across the country. So increasingly think about federation, allowing us to also do greater access to health data. Next slide. Some core principles. We have always been about international data sharing. It's where we aim. Increasingly, over the last 5 years, much more about translational. Technology hasn't been the barrier. I will say 90% of the time, the barriers that we've encountered have been about people, about cultures, about legal structures, about incentives. Technology seems able to catch up with whatever scale of data and complexity of data we can throw at it. More than anything, we've promoted interoperability, recognizing that there will be many solutions. Next slide. And you can build quickly through this one, Christina. It's just a little GA4GH ecosystem, 625 or so organizational members, thousands of subscribers, about 300 active volunteers, and a whole ecosystem. And that's, we created a very inclusive and a very big tent, and we've been delighted with the way the genomics community and increasingly others, industry and healthcare, have joined. Next slide. We have two fundamental building blocks for Global Alliance. The one on the left we own, it is us. We're a combination of 8 individual work streams, 6 are technical, 2 are foundational. We staff them, we organize them. They have volunteer co-chairs, and then they can have anywhere from 10 to 40 individuals collaborating, often on a volunteer or an assigned basis. We don't have any data. The data, the needs, the requirements, the applications, the implementations, they all take place on the right-hand side of this slide in our driver projects, of which there are 24 at the moment. And just a sense of scale, those 24 global driver projects alone represent about 3 million whole genomes. They're not all there, they're not all in place yet, but just the thought that we could go from one that cost $3 billion to the concept of being able to look at 3 million genomes in a very short time is actually remarkable. Next slide. These are the 24. Some of them are multifaceted. They are national health genomic medicine projects. They've got a rare disease component, complex disease, cancer. Some of them are pure cancer projects. Others are pure rare disease. I'm wearing one of my rare disease group t-shirts. And increasingly, there's a few in the areas of basic biology. And so it's a diverse group. It started off being very North American, European, and we've spent a lot of the last 2 years trying to make it more of a truly global cohort of driver projects. These 24 are our conduit to the other 200 plus, but it's not binary. We've got a lot of mechanisms and outreach in between. Next slide. So one of those things is from our 6 driver projects that are genomics in genomic medicine, we realize there are probably another 20 or 30 in the world who are not driver projects, but have got similar needs and requirements because they're doing genomic medicine in their country. And so we formed an implementation forum called Genomics and Health Implementation Forum, or GIF, and that's where our 6 come together as a core driving group, but they include this much broader collaboration and take the lessons learned at Genomics England, Australia, all of us, and apply them across the whole community of precision medicine, national medicine initiatives. Next slide. Beyond precision medicine, there are a ton of people doing genomics at scale, whether that's the international population cohorts in the IHCC or the disease cohorts in ICDA. HDR UK is now leading a new COVID initiative that's a global COVID initiative. And so what we've tended to do is say we won't compete with anybody. There is enough work and enough need for collaboration. We can't do it alone, even in the space of genomics. So let's collaborate and build a community of like-minded people. Next slide. As we move into healthcare, though, that nice, friendly, big tent of genomic researchers working together doesn't cut it when we move into healthcare. It's a much more structured environment, much more contracted, much more legal, with huge concerns, and things like accreditation. And so we need then to bring these global alliance standards alongside HL7, HL7 FHIR, ISO standards, CDIS standards, because the people doing clinical trials in hospitals or the people doing health informatics have already got standards groups. They don't need another standard. They need to know that the standards they're using are compatible and interoperable with these genomic research standards. So a lot of my time goes into these kind of external collaborations. Next slide. This one is busy, but the next slide is even busier. This is what I was talking about, of seeing it more as a learning health system, a continuum of knowledge generation from a patient and a data donor, all the way through a healthcare system back into research, and a virtuous cycle of knowledge. Historically, it takes about 17 years for a research finding to make it into routine healthcare practice. Even a good, solid research finding still takes 15, 17 years. It's too long and it's too ad hoc. So we're looking to see how we can play a role. Next slide. And this one just maps all the global alliance standards that we've been developing along those access points of that data continuum. So I'm not going to go through it. Guillaume will talk about a few of these standards and the way that they've been used in his environment. But generally, think about the system we're designing for, and then be specific about where we can make a difference. Next slide. I'm going to touch very briefly, just one slide, on public willingness to share data. So you can either look at this and say that in some countries, only 20% of people are willing to share the genomic and health data with for-profit researchers. But that rises all the way through to 60% in some countries willing to share it with their medical providers. So somewhere between 20 and 60%. And what's important is that we never, ever abuse or disuse or destroy that trust. But the key thing is, when I look at the 21 countries in this survey, 20% of 3 billion is a level of genomic sequences that we couldn't imagine actually completing in the next decade. It's way more humans are willing to share the genomic and health data than we could consume in the next decade. So I think the important thing here is not that we're going to be starved of people not trusting us, but we need to increase that trust, increase public awareness, so that we build this for the future. Next slide. So I think I've already talked about this, but the key for us is it would have been very easy to build an Anglo alliance for the top third of that pie for research. We could have easily, quickly done an international alliance of the major countries doing genomic research. The complexity, but the opportunity has been to build all of this way more difficult, and we were stops and starts, and it's not always straightforward. But the big win here is this will be true interoperability across all sectors. Next slide. Just one on COVID. I don't think I've done a presentation in the last 8 months without talking about COVID. It's been both a challenge, a massive distraction. A lot of our people actually been stopped in their tracks and having to pivot into new research. But generally, there couldn't have been a stronger use case for the need to be able to share health data, to be able to share public health data, to be able to combine that with genomic data and look at it at scale. And so in many ways, this has been, although it's been a challenge to our normal processes, it's actually made the best possible use case for why we do the work we do. G4GH has been modeled by a couple of groups in the COVID space, saying we need to start up quickly and we need to build a big tent and get everybody moving. How did you do it? The other one is we've had much better participation in our plenary meetings, double the number of people, double the number of countries represented. And I think we're all learning to use Zoom despite issues of bandwidth and backgrounds and all those kind of things. Next slide. I think that is probably my last one. It was. Thank you.
Thank you so much, Peter. We'll pass it over to you then, Guillaume.
Perfect. Thank you. So when Mark invited Peter and I to talk about data organization and sharing in genomics, I think we both said yes very fast, because as you will see in the presentation, what we do is very complementary and actually fits as an example of what Peter was describing in the GA4GH environment. So I'll follow up the presentation from Peter that really sort of provides the framework in which we're working, and give you a bit more of a historical perspective, basically, and show you as a genomic researcher how I've moved into that model myself for some of the projects that I'm involved with. So yes, as was mentioned in the introduction, I work at the McGill Genome Center. And if I can have the next slide. So, excuse me. I'm also responsible for this Canadian Centre for Computational Genomics. So this is a platform funded by Genome Canada that I run with my collaborator, Mike Bruno, in Toronto, that's meant to provide bioinformatics analysis, high performance computing services, and also software solution quite broadly in the life sciences. Next slide. So we support a whole range of projects, and I'm not going to talk about many of them today, but you can just see that it touches on cancer, infection, and immunity. So there's some interesting projects on COVID-19 that have been set up that Peter mentioned briefly that I think are quite relevant, obviously, this year. But what I'll focus on is really 3 projects to give you a sense of what we do in the context of data sharing in genomics. I'll talk about IEC in detail, and then briefly 2 projects, CanDIG and EpiShare, which are GA4GH driver projects that Peter introduced in the previous presentation. So next slide. So I know that this is a very diverse audience, so I'm not going to give you too much detail, but just a bit of introduction in terms of epigenomics as something that's complementary to genomics. So just very briefly, of course, each of us has a genome, but if you look in each of our different cell type, obviously that genome is activated in very different ways. Blood cells are quite different from skin cells and so on, even though they have the same genome. So the way the same code basically gets activated in different cell or in reaction to the environment, for instance, can be described as the epigenome. So we can use the same type of sequencing technologies, not just to look at genetic variation and mutations, but also to look basically molecularly at what's happening in these cells and what genes are expressed and which region of the genome is accessible. Next slide. So Peter talked about a number of large international projects. The IEC, which is the International Human Epigenome Consortium, is one of these that's focusing on doing this type of mapping effort. So just like we want to map and catalog genetic variation in the human population, we also want to understand and map the epigenome of all the cells in the body. And that turns out to be quite a challenge. And so in order to do this, we're doing this by splitting the work across different projects across the world. So this particular project aims to characterize 1,000 different cell type with really sort of an in-depth understanding of what's happening in these cells, what genes are expressed, and so on. Next slide. So again, similar to what we've heard, you know, there's a lot of willingness for this this consortium to provide the data that comes out of it in the context of open science. But we also have to be a bit careful here because some of the underlying data, because it comes from different individuals, different patients that have contributed these samples, has to be controlled access because there's some privacy issues with, just like with genetic data, with epigenetic data, there is sensitivity in terms of some of the variant information that's in these data sets. So although all the data is made available, there's a portion that needs to be controlled access in the sense that people have to demonstrate that they're researchers and that they have a valid question and request access. And we also have some data, which is more summary data that can be made available to the consortium as a whole. I mean, to everyone in an open fashion. Next slide. So a slide before. Oh, okay, this one. I saw 2 slides, I got confused. So, but this one shows the approach that we've used as part of the consortium to deal with this. So you can see on this slide that there's multiple data production groups that are typically in different countries. Some of the data needs to be submitted to these access controlled repository. These have been long established in genomics, some in the US, some in Europe, dbGaP and EGA, and even one here in Japan. And the data also gets processed, and a portion of the data gets released in a portal in a fully open fashion. But in order to access a lot of the underlying data, because it's sensitive data, users have to go through a DAC, a data access committee, to request access and access that underlying raw data. And next slide. So this, and as I mentioned, this I'm providing here a bit of a historical perspective. This is a project that was launched about 8 years ago, I believe, or maybe a bit earlier than that. And the model I showed is really what was implemented at the time and led to the portal that we released already over 6 years ago. So, this is, I think, the standard model that was used in the field in genomics at a time, where the data that then can be released openly is released openly through a portal, but a lot of the underlying sensitive data, as I showed, gets submitted through various international archive. Next slide. So since we generated all of this data as a consortium, what we're doing is we're trying to analyze this data jointly. So the individual groups have generated the data, but now the project has moved on to trying to analyze all of this data and working together and then learn from it. So this is sort of the phase of the project that we're currently in, and we're calling this the IP Atlas project. So we have an integrative analysis working group that includes people from all of these countries. And what we're trying to do is actually trying to reprocess all of the data that's been generated by the different group. And again, re-release the data once now that it's been uniformly processed. So we're just trying to be a use case, I guess, of that very, I think, important data set that we've generated. Next slide. But in the process, what we've realized is there's quite a number of challenges. So even though as a consortium, we're all part of the same group and we all had this ambition of creating a very useful resource, we realized that for us to actually be able to use the data, to combine data, we have to go through these data access committee ourselves to basically obtain access to the other data sets. We've also realized that these are very large data sets, and retrieving the data that others as part of the consortium have submitted, it's turned out to be quite a challenge. So we're talking here about, terabytes of data, maybe something of the order of 50 terabytes or something like that, that the individual countries have submitted, and trying to pull that data back, even though, again, so we're doing this amongst friends, but the paperwork and the logistics of doing that turns out to be quite a challenge. Next slide. So this is an analysis that Yan Jolie, also at the Center for Genomics and Policy at McGill, has done. So one of the things we did is to go back, again, even though all of these groups were part of the same consortium, each of them individually had slightly different conditions, because of course all this international consortium was actually a regrouping together existing projects, and some that were already ongoing and some new ones. But looking more specifically at the condition for using the data from these, in this case, 7 projects, we realized that there's some slight differences that end up being really problematic as we were trying to do the joint analysis and trying to retrieve all of this data together. So as you can see, there's only a few items like acknowledgements that are shared across all of the different groups, and then quite a bit of diversity in terms of condition, which makes our lives very complicated, even as we're trying to reanalyze this to be able to actually do that effectively. So next slide. So I use this just to highlight, this is a slide that you just seen from Peter, but this is really this transition really that we've seen. So the project was initiated in this older model of, yes, open research data, but using a shared centralized library, which ends up being quite challenging for some of these projects, especially if they involve controlled access and sensitive data. And so we also saw the need to start moving towards this new federated approach for the data set. Next slide. So one of the project that's a driver project that Peter mentioned is this EpiShare project, which is a project that's led and involves the people from this IEC consortium, but was a Genome Canada funded project here, where we're trying to revisit basically what we have done and try to see whether we could have done it better in a way that's more distributed, where I'm not going to go into the detail, but the idea is that instead of having each of these groups submit their data set to a common archive, we would really instantiate some of these federated nodes with the data such that each instance of the consortium could really have one of these nodes where they control the access. And then as a group, we would be able to share and analyze the data without having to go through this intermediate step of using a centralized repository. And this is, still sort of a test case and a work in progress. I don't know if it was clear from Peter's presentation, but a lot of these pieces are still moving in the sense that we are developing these standards jointly using these test cases. And it's not like we have this whole ecosystem fully up and running. Different portions of this at this stage are working, but we're really developing these standards using these driver projects basically to demonstrate what's needed or not. Next slide. So again, maybe not just a cartoon to illustrate this concept of a federated platform that hopefully will allow more fine-grained control directly by the data providers in terms of what they want to make available as part of their data. So we're hoping that here in Canada, and I'll get to that now, by using this type of strategy and working with Compute Canada and NDRIO, we're really going to be able to join the international effort of GA4GH. Next slide. So I briefly mentioned EpiShare, which is our version of trying to do this with epigenomic data. The second driver project that we're leading and that we're involved with is the CanDIG project. And this one is focusing on genetic data. Specifically, we're currently using it for a number of flagship projects in Canada. Signature is a project in Quebec, but we're working towards trying to use this platform for the Marathon of Hope, so to have an effective way of sharing cancer data that's going to be generated in Canada. So this software platform is really focusing on combining, as is mentioned at the bottom here, clinical and phenotypic metadata associated with sample and genomic variants, VCFs, and so on. And then we'll be adding to that. Next slide. So this is just a demonstration of this. But you can see here that on the right with the different provinces, the idea is that instead of really collecting all of this data into a single database, this is really demonstrating already 2 nodes that are sharing the data, and then you can provide a view of this federated data that's a unified view, but the underlying data remains in the location where the data was generated. And so this is, again, really where we're at now, with some components of that working and some other components that are still being worked on. Next slide. So I stole the second slide from Peter's deck just to highlight that, again, this complexity. I mean, starting into this, I didn't realize how complex this was going to be, right? So each step along the way requires, each of these little standards that you see on this slide requires extensive discussion with, a number of groups that have ongoing projects, and then reaching some kind of a consensus to reach an agreement of what's going to work for the majority, right? And then once that process is done, then the software platform and the implementation need to incorporate these standards. But it's really, again, if you look just for consent, the way consent need to be captured in such a way that, the information will be able to be retrieved in a machine readable way, and will be compatible across this diversity of project. you know, it's again, I didn't anticipate the level of complexity, but also it's great to see the progress that we've made. And, you know, Peter could answer this question, but I think 2 years ago we had no standard. And at this point, we have all the ones that you see here. And so it's quite impressive, the progress. Next slide. So the last few slides that I have in the deck is really to talk about sort of implementation of this type of approach in Canada specifically for genomic. So the vision again is to move away. Peter mentioned it in his presentation. We currently have silos that are maybe sub-provincial, as he mentioned, not even provincial. But how can we move away from that and really have an ecosystem in Canada that talks to each other? Well, I think the strategy is really to implement these international standards, because if we do that, not only will it be able to get things to talk to each other within Canada, but also to talk to these different international projects. Next slide. So this is a project that we're working on. So this is the culmination, I guess, of a number of projects. But currently we're working on really building this type of, well, I guess we didn't talk about that so much, but there's also a big concern on cybersecurity and how the data is actually hosted, because we're talking about genomic data and patient data. So there's a big component of this where not only does the infrastructure need to be interoperable, it also needs to be secure. So we're currently working on this project that really is going to be linking 2 nodes initially between Quebec and Ontario. So the group in Ontario is led by Mike Bruno and really builds on HPC4 Health, if you've heard about this. So this is a project that's setting up a secure environment for hospital data in Ontario. So we're now building a similar secure node in Quebec, and we're going to be linking those 2 nodes. Next slide. So the idea is, again, so on the left, you have the HPC4 Health node in Ontario. On the right, you have the Quebec node that we're setting up at Calco, Quebec. And the idea is really to set up these nodes in such a way that they can support a number of genomic projects using the GA4GH APIs, and again, to enable this interoperability. Next slide. So this is my last slide on this, but we view this as just the beginning, actually, of a network of GA4GH nodes, enabled nodes in Canada that are going to be building using a lot of the software infrastructure that we're building as part of CanDIG, EpiShare, and a whole other list of projects that our team has been involved with. And the idea is that with that, we will be supporting a number of key genomic projects in Canada. I mentioned the Marathon of Hope, but one that's, I think, very active and relevant, and this also goes to what Peter was talking about, is there is an effort in Canada, a national effort to sequence COVID patient and associated virus sequencing. And so this infrastructure ideally will also be able to support this project that was launched just this year, which is why it's not on my slide, because I haven't updated it yet. But next slide, and I think that's my final slide. Just to say, so this is work from a lot of people in my team, but at the bottom of the slide is also a very important slide that just shows some of the partners, and in particular, more recently, Calco, Quebec. So a lot of the work on building this secure node for health data is really joint work with Calco, Quebec. And I'm very excited to see sort of the next steps of that project. So thank you.
Thank you very much, Guillaume. Thank you, Peter. Really interesting and complex landscape that you're tackling at the moment. I'm wondering if we have some questions, Christina, to respond to at the moment.
We do. Okay. question. Most used anonymization tools and quality of results. Can anyone comment on that?
You're muted, Peter.
Peter, you're muted.
I'll do the big generalist answer from Global Alliance, and Gail might put some depth to it. Certainly our data security group has put a lot of effort, and it is coming out of leadership work from EPFL in Switzerland on things like homomorphic encryption, multi-party computation, the sophisticated ways of encrypting data. I think in many countries there's been anonymization, pseudo-anonymization, but increasingly I think people are recognizing that some of this data, it's important to keep the identity, keep the linkage. It's health data. If you anonymize it, it's really important that you don't lose the value of that data. And so there's been an equal amount of effort, perhaps more, on secure clouds, on secure environments, on trust-based relationships, then turned into technical solutions. So I think what people are recognizing, in the old days, we could know the individual. They were in a consortium. We knew where they worked, who they were, and we could trust them to get a copy of the data. Increasingly, it's, I will give you permission to come into my data. But since that could be thousands of researchers from inside my consortium or more broadly, how can I verify identity, and how can I make sure that data use is correct? And so we've got one group called Jury that is focused on building a data use ontology. So very clear clarity on the data use conditions, machine readable, and then researcher identity is represented as a passport to which you will build visas. And so therefore this validation of my machine ready identity and my machine ready, what am I going to do with the data? It probably will not replace every DAC on Earth, but it will give those DACs much better tools to expedite thousands of requests. And if you've been permissioned in one environment, it should accelerate your permissioning in a different environment that has reciprocal agreements regarding the validity of that visa or of that passport with those visas in it. So I would say that although there's a lot of work on anonymization, I can't actually give a how well are they working, but I would say that more of the effort and energy has gone into being able to create better tools to clarify who's the researcher, what are they going to do with it, and then audit tools so that when they came and behaved inside your environment, they actually did what they said they would do. What we haven't had to deal with yet, fortunately, I don't know of anybody who's had to take away somebody's passport for bad behavior or discipline people for messing up the environment. But I think increasingly people are more worried. In the beginning, the emphasis was on privacy. More recently, it's been on people stealing data, corrupting data, ransoming data. So it's all the general issues of cybersecurity have become much more in our discussions, whereas 5 years ago, it was essentially about privacy. Security meant privacy. Guillaume?
Well, I don't have a whole lot more to add. I mean, I agree with you that if you think, I mean, not just health data and linkage, but even genetic data, we've realized that it's very difficult to hide the identity in this sort of a moving target, because even if we try, people realize that actually there's still a way to recover, right? So it becomes you know, a bit futile to try to anonymize genetic data, is what, unless you do it to the point where it's no longer useful, is what people have realized. So there's a movement towards what Peter described, which is much more in terms of, you know, doing the authorization, identifying who's going to be doing that, who's accessing the data, and making sure that they have the rights to do what they're planning to do.
And I would say we've also looked at the type of data, and wherever possible, if it is non-identifying and it can be shared publicly, put it in a public data in the easiest. I can now look up world-class interpretations of significance for BRCA1 and BRCA2 from my smartphone, and triple the number of variants that were there 7 years ago, and almost a third of them expert curated from my phone. and I could be anywhere in the world and do that. But that was because it wasn't it's not case level data. It's not identifying a human being. It's sharing what we know about the impact of those variants. So I think that's been the other thing is think about tiered access. If it's public and shareable, then put it on the web, get it out there as fast and as broadly and with as high utility as possible. If it's highly sensitive, either because of vulnerable populations or specific individuals, put it in the most controlled environment. And then sometimes we have a layer in between, which is registered access. It's not the most confidential data, but equally we want to know who's accessing it, how frequently. And so we have registered access in some cases. So okay, I'll stop so we can get to something else.
Fantastic. I see there's a question here. What is used for authentication and authorization between the Quebec and Ontario nodes?
So I can probably take that one. So this is actually an example of one of these moving targets that I mentioned. I know that initially in our solution, we were using, I believe, Keycloak or some different protocols, but I know that this is being decided at the level again of GA4GH in terms of what is the standard that's going to be used across all of these projects, and we're currently working towards adopting those standards. So I unfortunately don't have the answer, but I would encourage you to look on GA4GH to see exactly what standard for authentication is now being recommended, unless you know, Peter, but I
was going to say we've relied. So in addition to our driver projects, we've got a couple of big data generators, owners, funders, and we tend to lean very heavily into them. And if we can get them to agree on something, we know it brings a huge swath of world pressure with it. And that's EBI and Elixir in Europe. So the EMBL EBI and Elixir for the European infrastructure. And then there are other places, NIH and things like NCBI. So I think sometimes they can get on the same page, and other times they have, because of all their legacy systems and approaches, or the fact that one's a loose federation of institutes and the other's much more tight. That's where most of our AAI is coming from. It's where much of the thinking into passports went in, what kind of tokens can be used. And people had already started, and so somebody's got to compromise a little bit. Or we say, actually, you can keep using yours for your purpose, and we'll adopt this over here, and we'll ensure that the two are interoperable. So I'm not exactly sure where we're at, but I can say that most of the leadership has come either from the Elixir EBI group on the Hingston campus or from NIH.
I think we have actually a response, if I'm not mistaken, to this, and it's from David Rottenberg, who notes that we're currently leveraging distributed graph databases with standardized interoperable schemas to perform federated queries for neuroimaging and clinical data across a network of nodes. How are you envisioning the technical scaling of analytics across node-based distributed networks for increasingly large data sets?
Okay, I'll give the generalist overview and then let Guillaume give the details. So when we both showed that chart, this linkage of standards, and I will say in the first 2 or 3 years, we produced a fair amount of standards, a little bit more on some like regulatory and ethics, but nothing like the output that we've done in the last 2 to 3 years. But they were all pretty much individual standards. We hadn't put a lot of time and effort into how these standards can work together. Whereas this year, more of our effort is, rather than generating new standards, it's creating systems where they can work together. And most of our energy at the moment is on a thing we call FASP, which is the Federated Analysis Systems Platform. And it's linking a number of these standards, particularly the cloud-based standards, and saying, how can we put 3, 4 of these together and then operate on data, either different data in the same environment, same data in different environments, or the more complex case of different types of data in different environments. And one of the places we've said is we really need to start thinking about is how do we align genomic data with imaging data? because there's some real richness in that. And places like the Cancer Commons that's hosted at University of Chicago actually is now co-hosting genomic data and imaging data from a cancer perspective. And the question is, where do you get not just have them in the same repository, but how do you start to create some real exchange between those two disparate forms of data? Guillaume, over to you.
Well, I was just going to say, I think there's a lot of parallels, I think, in genomics and in other fields, I suspect, in imaging, where the same types of strategies are being used, right? So how do you federate the analysis of these large data sets? And most of the effort initially was in making sure that we could package the pipelines such that we could rerun the same tools in this type of distributed environment. But the challenge was to realize that, the compute environment ended up also being quite different. So standards was also required to enable that. So it's a lot of the time ended up being spent in just making sure that these simple pipeline just for genomics could be migrated to this type of distributed environment where all of these questions of authentication and access were there, and then specificities of the different tools and the different environment where these tools have, were being run. But that said, I think, a lot of the work that's been done in genomics or is being done in imaging, I think the last point that Peter mentioned, the way these approaches are going to be reconciled such that they're also compatible across multiple data types, right? So obviously we don't want to build a federated model for genomics that's not going to be compatible with the similar federated model potentially for imaging, right? So I don't know at what stage this is going to become a top priority for the Global Alliance, but I suspect it will become increasingly important indeed.
So it's actually becoming more important locally. So I'm seeing the bringing of these things together. Maybe it's because I'm based at OICR and we're near neighbors with Ontario Brain Institute. So that's one reference point. Within the Marathon of Hope, you've got the CanDIG platform that Guillaume was describing, but you've also got Imagia and its AI capabilities that I think were built in the imaging world. So I think we're also seeing in Canada, as we start to build this more local federated system to share Canadian health data, Canadian genomic data, I think it's going to be one of the environments where we'll also try and bring in other forms of research health data. And it wouldn't surprise me at all if imaging data is the first to line up alongside genomic data.
I'll throw, Patrick Matlock has a few questions here. I'm going to throw a couple that seem to be interconnected here at you. APIs were mentioned. The norm today at most universities is to use SAML 2.0 authentication and authorizations, which is defined to not work with APIs. So how will the APIs be authenticated and authorized? And then he a little later goes on to say, FYI, OpenID Connect solves both the federated authentication and authorization and supports APIs. It supports Caribou, SAML2, I don't know this one, OAuth2 okay, thank you. Hadoop clouds and support this, for example. Have you looked at it?
Again, big picture for me, yes. In fact, wherever something is already well established and it's used in many different environments, we've tended to adopt or adapt to ours. So OAuth2 has been a big component of this from the beginning. And so that another key principle, wherever there is something that is already well established and can be adapted, don't reinvent the wheel. Don't make something specific to genomics. So use the tools that are available wherever they are. Often what we're doing is making those tools, those approaches well known, because you can get very siloed inside your environment and think all you've ever done is the things that are local to your environment. The minute you move into a cloud-based environment, then a whole bunch of things. So if something's already working, we don't tend to try and reinvent the wheel. Guillaume, any?
Well, I don't have much more to add. So again, I haven't followed these discussions in as much detail, but this is an example of what I was describing, where the different projects are bringing their constraint and their suggestions and the system that they're working on. And the work of these working streams is to come up with some kind of consensus that will be as compatible with, you know, all of these projects and all of these dependencies as much as possible. So for a more precise answer, I would really encourage you to look at the documentation of what is being recommended as part of these work streams.
And the two work streams to focus on, both because looking at the standards that have been released will tell you where we are today. Looking at what's in the work streams and the roadmap will tell us what we're working on at the moment. So the two that will be most relevant to this are the DURI, Data Use Researcher Identity work stream, and one of the foundational work streams, which is the data security work stream. So those are the two where most of these conversations, and in fact, at one point they were starting to take different approaches, and we had to bring them back together and say we do not want to go left, you know, go down this path with our data security group and go down this path with our researcher identity group. So they're the two places to look for it. I will say a lot of this often then goes back with technical developers saying, but we need to know what the policy is. We need to know what the ethics are saying. We need to know what the consents were, because that creates the environment, and then we can use our tools. So.
I see we're running low on time here. I think, Christina, I just want to check in with you. Do we have time for one more question, or are we at the end?
I mean, it's up to your availability. We are at 1:59.
Gentlemen?
Give us one more.
All right. What can be done to address the fragmented requirements on data by consortium members and help to promote open data sharing?
It's really interesting because some of this, I see some very loose consortia being built, and they've gone loose so they can include, and the International 100K Cohort Consortium is a great example. We feel we need to spend a few years building a community and creating a more level field, disseminating tools and best practices, and bringing everybody up. Within that, potentially 100 different cohorts in a big consortium, I think when 6 of them get together and they want to do a joint research project and they need to really start giving each other reciprocal access, then they'll form tighter rules and conditions. But in the first place, if we put too many rules and conditions, we wouldn't be able to create the big consortium, the learning environment. We talked about federated access, and you can do things within a tight consortium, like the International Cancer Genome Consortium was fairly tight. But the Matchmaker project we have is actually 6 originally, now 8, very different PI-driven researcher, rare disease matchmaker databases that are not, it's a very different kind of federation. You go into the one of your choice, it searches the others for you and says, we don't have a result, but there is one in Australia. And then you switch into very manual processes to follow up. But it's a good way of finding the data. So it's federating the finding part of the data. It's very different from the granting access, which is different again from doing the analysis. And I think sometimes we've got to say, right here, right now, let's focus on that layer, and then we can start to build all the trust and the conditions that will enable full analytic access.
Maybe the only small thing I would add to that is, to me, the other thing that's going to help. Of course, if we're working with existing projects, it can be a challenge. But I think as we move forward and we establish what are the requirements that need to be part of consent forms for this to be for these studies to be part of a GA4GH type of environment, and those can be included in new projects being set up, I think this will also help quite a bit going forward. There's a lot of projects that are just saying, you know, what needs to be included, and unless we can provide information on these requirements, you know, we're sort of running behind again in terms of later on.
Thank you so much. You got to have the last word there, Guillaume. Thank you both, and thanks to all of our participants. Katrina, do you have any other, Christina, sorry, do you have any other closing remarks at this point?
You covered it all. We'll be sending out the recording and the slide decks in the next couple of days.
Thanks everybody for attending, and thanks to our wonderful speakers. Take care.
Thank you.
Goodbye.
All right, bye.
Bye.
Bye-bye.